thank you all for being here. Um, just a quick introduction of kind of what we'll be covering today, and uh, introduce myself and my co-moderator Shah Hussein, who's with us today. So really, I'm David Lafon. I'm a translational physician scientist at UAB, University of Alabama in Birmingham, and I work mainly in COPD, looking at immune responses to respiratory bacterial infections in COPD. And so I work with a couple COPD kind of cohorts, including spiromics and COPD gene. And every time I go to a COPD meeting or go to ATS, I'm always blown away by all the different techniques. I think we all kind of have our head down working on in our own area. But then when you kind of hear what everyone else is doing and what everyone's up to is just incredible. And I always come away from those experiences with a lot of ideas and just an admiration for what folks are doing. So with that in mind, we wanted to kind of highlight some techniques in COPD research, and this will hopefully be a little bit of an appetizer for ATS, which is rapidly approaching, to kind of get some wheels turning and give you some ideas for what folks are up to in advance of the meeting. And so, without further ado, I'll let my co-moderator Shah introduce himself as well in just a second. We will, in terms of the format today, we're going to go give each of our presenters a chance to present a couple slides introducing and discussing their technique. And then we'll have time for one or two questions after each talk, and then hopefully have some time for any further questions at the end. So we're going to be keeping an eye on the chat. So please feel free to drop questions in the chat or raise your hand if you have a question for any of our presenters. And thank you all for joining us, and special thanks to all our guest presenters today as well. So Shah, I'll let you introduce yourself, and then we will get started with our first talk.
Yeah, thank you, David. Like it's a nice way to present. And then my name is Shah Hussein. I'm assistant professor at University of Alabama, and my work is mostly related to how the small airway disease happening, or like how it initiate like a COPD, emphysema, or genetic, what are the genetic cause of the disease, like for alpha antitrypsin deficiency. So basically, I'm using the different kind of tools and the model system like a ferret as like a to answer the question of how the airway loss is happening and how it is happening. So I'm going to talk about in my work in my lab is basically related to this airway loss and how senescence works in the airway loss. So if you go ahead and then later on, like you will see that I'm going to talk about how I'm using the model and animal model ferret, which is one of the hot model, excellent model for the study of airway disease biology, COPD and emphysema. So I'll talk about the later, like, but for a timing, my work is related to basically small airway disease. So that's the one thing. Thank you so much. So later on, we can move it with the Krish Genschmer, who is like a associate professor at the University of Alabama, and he is gonna. I think David can gonna introduce him to like what he's doing.
Sure.
David.
Yeah. So Chris Genschmer is an associate professor at UAB, and he did his PhD in microbiology at UAB, and then stayed here for a postdoc, where he began studying the role of extracellular vesicles in COPD and in smoking related inflammation and neutrophilic inflammation. So Chris has done a lot of really interesting work in this area, and I think found an even broader role for these exosomes, which I'm sure he'll cover in his talk. But I also have to give Chris a shout out for his role in kind of professional education here. So a few years ago, Chris had the idea to kind of start up a techniques-based seminar series here at UAB, and where we have a monthly seminar where we cover some pulmonary research techniques. And so I got to give him credit because that was kind of the inspiration or partial inspiration for why we wanted to do kind of a lightning round style talk and do what we're doing here today. So I'll turn it over to Chris.
Thanks, David. Yeah, so I work with extracellular vesicles real briefly, for those who don't know what an EV is, or exosome or extracellular vesicle, they're just tiny 100 nanometer sized vesicles that all cells secrete of some form or fashion. Specifically, I look at neutrophil-derived extracellular vesicles, and the reason for that is because they tend to be covered in neutrophil elastase. So our rationale for looking at these is that we know that neutrophil elastase-driven alveolar destruction can persist in a COPD lung, even when there's abundant alpha-1 antitrypsin. And what we've discovered is that elastase on the surface of these extracellular vesicles are resistant to alpha-1 antitrypsin. So when there's elastases on these extracellular vesicles, the person who has these in their lung are basically alpha-1 antitrypsin deficient, at least with respect to this elastase that's on those extracellular vesicles. We found that these EVs can be detected in patient BALF, they can be detected in the sputum, and even exhale breath condensate. And this kind of offers a real-time snapshot as to some sort of the active pulmonary proteolysis that these enzymes can be doing inside the lung. And these EVs represent a novel therapeutic target, actually, because we're beginning to look at methods to dissociate this elastase from the surface of these extracellular vesicles, and that through ionic disruption. And that, once again, allows them to be inhibited by alpha-1 antitrypsin. So it restores sort of the homeostasis and the protease-antiprotease balance. But real quick background as to why we looked at these. We started looking at activated peripheral blood neutrophils that we'd get from donors. And after we would stimulate them to degranulate, the extracellular vesicles that were that we captured from this process, we put those into a mouse model of emphysema. So we basically administered them intratracheally. And in the top panel here, you can see mice that were given PBS or EVs from quiescent peripheral neutrophils had no effect. But those that were dotted with this neutrophil elastase, you could see the alveolar destruction and alveolar enlargement. And so that made us want to look at these patient samples that we had, this cohort of patient samples that we would have, whether they're never smokers or COPD patients. And we discovered that the extracellular vesicles from the BALF and the sputum of these patients had a very similar phenotype. If we were to give mice PBS again, we'd see no change. EVs from never smokers, we didn't see a very large amount of alveolar destruction. But those that came from patients with COPD had this very high elastase presence on the extracellular vesicles. So this made us want to design this sort of mouse model of emphysema using extracellular vesicles to sort of gauge and measure the potential of these extracellular vesicles that are happening within this patient. Can you go to the next slide, please? So basically how we do this is we'll typically get either BALF, sputum, or exhale breath condensate sample, and we'll use differential ultracentrifugation to isolate these extracellular vesicles. With that, depending on the amount that we have, we can look at the surface elastase activity. We can either look at that through a fluorogenic substrate, or we can even do some flow cytometry to measure elastase on the surface of these extracellular vesicles. And we typically count the numbers and concentrations of these using nano tracking analysis or spectrodynes. But really what we like to do is we like to look at the emphysematous damage that's assessed in these mice. And we do that with H&E staining. We calculate the mean linear intercept. And just so you can see some of this unpublished data that we have here, based on the patient's lung pulmonary dysfunction, the EVs that come out of this patient cause an increased amount of alveolar damage in the mouse. So COPD patients who are current smokers have a much higher a burden of disease that are given to this mouse with these extracellular vesicles. So we're basically measuring the pulmonary dysfunction in these patients using these extracellular vesicles. And I can tell you that we have begun to see a correlation between FEV1 readouts of the patient inversely to the amount of damage that they can do in a mouse lung. And that's quite interesting for us now. We also have generated a mouse model where we can smoke mice for 1, 3, or 6 months and take the EVs out of that and see how smoking directly impacts the generation of these extracellular vesicles. So our future directions here is we really just want to look at these EVs from patients who mainly have quit smoking, and can we detect a turning point to where we might be able to instill some sort of strategy to arrest the progression towards COPD progression in these patients who have quit smoking, and also look at some therapeutic strategies that might be able to reduce and release elastase from these extracellular vesicles. That's it. I've got time for some questions, I believe.
Yes, please drop any questions in the chat or go ahead and feel free to unmute and ask or raise your hand, however you prefer. We got a couple minutes for questions from Chris. All right, looks like about it. Go ahead and feel free to ask your question.
Hi, can you hear me?
Yeah.
Yep.
Fascinating. Are you planning to pursue like maybe proteomics or kind of to figure out what else is in these extracellular vesicles?
Yes, we've looked at the proteomics. It's not just elastase that's on there. You've got MPO, PR3, various other proteases that come from neutrophils, and basically anything that's in granules during this time can be there. We're also looking at macrophage EVs. But the reason we focus on elastase is because of that specific sort of phenotype that when elastase is on these EVs, they are effectively alpha-1 antitrypsin resistant. And so that looked like a major therapeutic aspect that we could manipulate maybe to cause these to be less pathogenic. But yes, we have done the proteomics and we do see an increase in a lot of those proteases, especially the serine proteases that you would expect.
The reason I ask because there's a new like cathepsin C inhibitor that is in bronchiectasis, but it'll hopefully be extended to like COPD and like maybe even cystic fibrosis. And this would be a wonderful kind of way to assess response to these like neutrophil kind of extracellular enzyme inhibitors.
Oh, absolutely. That's you said cathepsin C?
Yes, correct.
Yep, I'll have to look into that. Thank you.